Hello, and welcome to Go Beyond A&P. When most people think about vaccines, they picture a simple injection, a quick shot in the arm, maybe a sore shoulder for a day or two. But what most people never see is the extraordinary biological journey that begins the moment the needle leaves the arm. In today's episode of Go Beyond A&P, we're going to follow that journey step by step. We'll begin in the deltoid muscle where the vaccine is injected. From there, we'll travel through the lymphatic vessels into a nearby lymph node where the immune system begins training itself to recognize a future threat. Finally, we'll end our journey deep inside the bone marrow where specialized immune cells can survive for decades, quietly waiting for the day they may be needed. Before we begin, let's define a few terms. An antigen is any molecule that the immune system can recognize as foreign. In vaccines, the antigen is a harmless target that teaches your immune system what the real pathogen looks like. An immune response is the coordinated series of events your body uses to recognize, attack, and remember that antigen. And finally, immunological memory is the ability of the immune system to respond much faster and more effectively the next time it encounters that same antigen. It's this remarkable memory that makes vaccines work. One thing I'd like you to notice throughout today's presentation is a timeline shown across the bottom of this slide. Unlike many immune system lectures, we're not just going to learn what happens, we're also going to learn where it happens and when it happens. So by the end of this video, you'll see that vaccination isn't a single event. It's a carefully choreographed journey through the body, transforming a tiny amount of harmless antigen into protection that can last for years or even a lifetime. So let's begin where every vaccination begins, the moment the needle enters the deltoid muscle. So every vaccine begins with a very small injury. This may sound surprising, but it's actually an important part of the process. As the needle passes through the skin, it travels through the epidermis, the dermis, and the subcutaneous tissue before reaching the deltoid muscle where the vaccine is deposited. Whether the vaccine contains an inactivated virus, a purified protein, a viral vector, or messenger RNA packaged inside lipid nanoparticles, the goal is the same. Deliver the antigen, or the instructions for making that antigen, to local tissues where the immune system can begin its work. So notice the two panels on the slide. On the left, you can see the anatomy of the injection itself, and on the right, we've zoomed in to the microscopic level. Immediately after the injection, the tissue responds to both the tiny mechanical injury caused by the needle and the vaccine components themselves. Within minutes, nearby blood vessels become more permeable, allowing immune cells to leave the bloodstream and enter the tissue. Among the very first responders are neutrophils, monocytes, and specialized immune cells called dendritic cells. Think of these cells as the body's first responders. They aren't yet launching a targeted attack. Instead, they're asking a simple question. What just happened here? This early response belongs to the innate immune system. Unlike the adaptive immune system, the innate immune system doesn't recognize specific pathogens. Instead, its job is to recognize signs that tissue has been injured or that something unusual has entered the body. Its job is to contain the situation, gather information, and prepare the adaptive immune system for what comes next. That preparation begins almost immediately. That mild soreness you often experience after a vaccination isn't the vaccine causing disease. It's evidence that your innate immune system has recognized something important and has begun recruiting immune cells to the injection site. So, how do these immune cells recognize that something deserves attention? The answer lies in an ancient surveillance system built into nearly every cell of the innate immune system. One of the most fascinating aspects of immunology is that your immune system doesn't simply recognize germs, it recognizes danger. The cells of the innate immune system are equipped with specialized receptors called pattern recognition receptors, or PRRs. You can think of PRRs as the immune system's burglar alarm. Instead of recognizing one specific virus or one specific bacterium, they recognize common molecular patterns that are associated with infection, cellular damage, or tissue stress. Different PRRs recognize different danger signals. Some detect viral DNA, others recognize bacterial cell wall components. Still, others detect molecules released by injured cells. And when these receptors are activated, they trigger a cascade of intracellular signaling that ultimately leads to the release of cytokines and type 1 interferons. So let's pause for a moment and define those terms. Cytokines are small signaling proteins that immune cells use to communicate with one another. They're like the text messages of the immune system. Interferons are specialized groups of cytokines that help establish an antiviral state, warning nearby cells and coordinating the early immune response. Now look at the comparison table on the right side of the slide. During the first several hours after vaccination, the innate immune response dominates. It occurs primarily at the injection site. Its purpose is immediate containment and communication. But over the next several days, something much more sophisticated begins: the adaptive immune response. Unlike the innate immune system, the adaptive immune system learns. It remembers, and it becomes more effective every time it encounters the same antigen. The bridge between these two systems is one of the most remarkable cells in immunology, the dendritic cell. So let's meet the cell that connects the innate immunity with adaptive immunity, the dendritic cell. If I had to choose one cell that deserves the title of teacher of the immune system, it would be the dendritic cell. Dendritic cells are professional antigen presenting cells, often abbreviated APCs. An antigen presenting cell is exactly what the name implies. Its job is to capture antigens, process them into smaller fragments, and display those fragments to T cells. So the adaptive immune system can learn what to recognize. Notice how the dendritic cell changes across this slide. On the left, it's functioning as a highly active scavenger. It constantly samples the environment, engulfing vaccine particles and foreign material through a process called endocytosis. Inside the cell, those proteins are broken into smaller peptide fragments. The dendritic cell then loads those fragments onto specialized proteins called major histocompatibility complex molecules, or MHC molecules. So think of MHC molecules as display cases. They hold tiny pieces of antigen on the cell surfaces so T cells can inspect them. And as this process continues, something remarkable happens. The dendritic cell undergoes a complete transformation. It reduces its phagocytic activity, dramatically increases expression of MHC molecules and co-stimulatory proteins such as CD80 and CD86, and prepares for an entirely new mission. It stops acting like a scavenger and starts acting like a teacher. But there's one problem: the students, the naive T cells. They're not here in the muscle. They're waiting somewhere else. To find them, our dendritic cell has to leave the injection site and begin its journey through the lymphatic system towards the nearest draining lymph node. Our dendritic cell has now completed its first job. It's captured the vaccine antigen, processed it into peptide fragments, and displayed those fragments on MHC molecules. But there's one problem. The immune cells capable of launching a highly specific response aren't located here in the muscle. They're waiting inside the draining lymph node. So how does the dendritic cell find them? The answer is the lymphatic system. Most people think of the lymphatic system simply as a network that drains excess fluid from the tissue. While that's certainly one of its jobs, it also functions as the immune system's transportation network. So notice the image on the left of the slide. The vaccine was injected into the deltoid muscle, but instead of entering a vein, our mature dendritic cells entered tiny lymphatic capillaries. These vessels collect fluid from surrounding tissues and carry it toward the axillary lymph nodes, which serve as a primary draining lymph node for the arm. Now look at the diagram on the right. Once the dendritic cell arrives, it doesn't randomly wander through the lymph node. It enters through the afferent lymphatic vessels, passes beneath the capsule into the subcapsular sinus, and then migrates into a specialized region called the paracortex. So think of the lymph node as a university. Different students attend different departments. B cells spend most of their time in the follicles. T cells gather in the paracortex. Our dendritic cells know exactly where to go because its students, the naive T cells, are waiting there. This remarkable organization isn't accidental. It dramatically increases the chances that a dendritic cell carrying the right antigen will encounter the one T cell capable of recognizing it. In immunology, location is everything. The lymph node isn't simply a filter. It's an extraordinarily organized meeting place where the innate and adaptive immune systems come together for the very first time. When physicians notice an enlarged lymph node after a vaccination, it's usually a sign that immune cells are actively interacting within the node, not that the vaccine has caused an infection. Our dendritic cell has arrived at the right location. Now it must perform the most important job of its life, activating the T cell. So this slide illustrates one of the most important conversations in all of immunology. Our mature dendritic cell has reached the paracortex of the lymph node, carrying antigen on its surface. Nearby, thousands of naive CD4 positive T cells are constantly circulating through the lymph node, each displaying a unique T cell receptor, or TCR. And here's something remarkable. Although millions of T cells pass by the dendritic cell, only a tiny fraction possesses a receptor capable of recognizing the displayed antigen. It's like trying thousands of different keys in a lock. Eventually, one key fits. When that happens, the dendritic cell delivers three essential signals. The first signal is the antigen recognition. The T cell receptor binds to the peptide presented on an MHC class II molecule. This tells the T cell exactly what antigen it has encountered. The second signal is called co-stimulation. Proteins such as CD80 and CD86 on the dendritic cell interact with the receptors on the T cell, confirming that the antigen represents a unique threat. Without this second signal, the T cell becomes inactive rather than activated. It's one of the immune system's most important safeguards against attacking harmless molecules or even our own tissues. The third signal comes from cytokines. These chemical messengers influence what type of helper T cell will ultimately develop. In this case, many activated CD4 positive T cells begin differentiating into T follicular helper cells, often abbreviated TFH cells. Notice the transcription factor shown on the right side of this slide, BCL6. Think of transcription factors as genetic switches. Once BCL6 is activated, it turns on an entire program that transforms a naive T cell into a TFH cell. So why is that important? Because TFH cells become the primary teachers of B cells. Without them, high-quality antibodies would never develop. Our immune response has now reached a new stage. The teachers have been trained. Now it's time to find the students that they were meant to teach. So patients with defects in TFH cell development often have difficulty producing high-affinity antibodies, even though their B cells are present. And this highlights how essential TFH cells are effective vaccine responses. So the newly activated TFH cells have one destination in mind, the border between the T cell zone and the B cell follicle. One of the many things that fascinates me about immunology is that immune cells don't simply bump into one another by chance. They're guided by an intricate system of chemical signals. And these signals are called chemokines. Chemokines are small proteins that act like molecular GPS signals, directing immune cells to very specific locations within lymphoid tissues. Look closely at the diagram. On the left is the paracortex, often called the T cell zone. On the right is called the follicle, which contains large numbers of naive B cells. These two populations normally remain separated, but after activation, something remarkable happens. The newly differentiated TFH cell begins expressing the chemokine receptor CXCR5. That receptor allows the cell to detect increasing concentrations of a chemokine called CXCL13, which is produced within the follicle. It's almost like following the scent of perfume through a crowded room. The stronger the signal becomes, the closer the TFH cell moves towards that follicle. At nearly the same time, B cells that have recognized the same antigen begin migrating in the opposite direction. Both cell types are independently following chemokine gradients. The result is that they meet at a very precise anatomical location called the TB border. This isn't simply convenient, it's essential. The adaptive immune response depends on these two cell types finding one another at exactly the right place and at exactly the right time. Their meeting will determine whether long-lasting immunity develops. And when these two cells finally meet, one of the most carefully choreographed interactions in immunology begins. So the title of this slide comes from a ballet. A pas de deux is a dance performed by two partners whose movements are perfectly coordinated. I think that's a beautiful analogy for what happens here. At the TB border, an activated TFH cell encounters a B cell that has recognized the very same antigen. This isn't a random encounter. It's a highly specific molecular conversation. So notice the proteins connecting the two cells. First, adhesion molecules help the cells remain physically attracted long enough for communication to occur. Next comes the interaction between CD40 on the B cell and CD40 ligand on CD40L on the TFH cell. This is one of the most important survival signals in adaptive immunity. Without it, the B cell can't continue developing normally. Finally, the TFH cell begins secreting cytokines, including IL-21 and IL-4. These cytokines tell the B cell what to do next. They stimulate rapid proliferation, initiate class switching, allowing the B cells to produce antibodies other than IgM, and prepare the cell to enter the germinal center. So let's define class switching for a moment. When a naive B cell is first activated, it usually produces IgM antibodies. Remember, Ig is immunoglobulin. As the immune system matures, those same B cells switch to producing IgG, IgA, or IgE antibodies without changing what antigen they recognize. In other words, the antibody keeps the same target but acquires new biological capabilities. By the time this conversation ends, the B cell has received everything it needs to continue its journey. Now, it enters one of the most remarkable structures in the immune system, the germinal center. This is where evolution happens on a microscopic scale. Individuals with inherited CD40L deficiency cannot generate normal germinal centers or effective class switching. And as a result, they have profound difficulty producing high-affinity IgG antibodies despite having a normal numbers of B cells. All right, inside the germinal centers, B cells enter an intense period of mutation, competition, and selection, a process that ultimately produces the highest quality of antibodies your immune system can make. So we've arrived at what I consider one of the most extraordinary structures in all of immunology, the germinal center. If you have never heard of a germinal center before, don't worry. It's simply a temporary structure that forms inside of a lymph node after a strong immune response begins. Its purpose is remarkably simple. Take good antibodies and make them even better. Notice the germinal center is divided into two distinct regions, the dark zone and the light zone. Each has a very different job. So let's begin in the dark zone. Here, activated B cells begin dividing at an astonishing rate. Some of the fastest dividing cells in the human body are found inside germinal centers. But something else is happening at the same time. The genes responsible for producing antibodies are deliberately mutated. Yeah, you heard that correctly. Your immune system intentionally induces mutations into its own antibody genes. This process is called somatic hypermutation. So let's define that term. Somatic means that the mutations occur only in these immune cells. They are not inherited and they can't be passed on to your children. Hypermutation means these mutations occur at a rate millions of times higher than normal mutation rates found elsewhere in the genome. So why would the immune system intentionally create mutations? Because every mutation produces an antibody that's slightly different. Some bind the antigen worse, some bind about the same, and a few bind much better. So those newly mutated B cells then move into the light zone. This is where the real competition begins. Displayed through the light zone are tiny amounts of antigen held by specialized cells called follicular dendritic cells, or FDCs. But don't confuse these with the dendritic cells we talked about earlier. Despite their names, they have completely different jobs. Follicular dendritic cells don't activate T cells. Instead, they act like judges in a competition. Each B cell must capture an antigen from the FDCs and then present it to the nearby TFH cells. Only those B cells with the highest affinity antibodies can effectively capture enough antigen to survive. The others undergo apoptosis or programmed cell death. That's exactly what the funnel on the right side of the slide represents. Many cells enter, very few cells survive. Their survivors aren't chosen by chance. They're chosen because they bind antigen better than their competitors. This process is called affinity maturation. By the end of the selection process, the immune system has transformed a modest antibody response into one capable of recognizing its threat with remarkable precision. Evolution usually takes thousands of generations. Your immune system accomplishes a miniature version of evolution in just a few days. One of the reasons booster vaccines are so effective is that they send memory B cells back into germinal centers, allowing additional rounds of somatic hypermutation and affinity mutation. Each exposure gives the immune system another opportunity to produce even better antibodies. So, what happens to the elite B cells that survive this rigorous selection process? Their paths begin to diverge. So at this point, our B cells have completed one of the most demanding selection processes in the immune system. Now they face another important decision. What should they become? The answer depends largely on the quantity of the help they received from TFH cells during the germinal center reaction. Some B cells differentiate into short-lived plasma cells. Others become long-lived plasma cells. Although both produce antibodies, their roles are quite different. So let's start with the short-lived plasma cells. These cells remain within the lymph node, particularly the medullary cords. Think of them as the emergency response team. Within days, they begin producing large quantities of antibodies, creating a rapid spike in antibody levels that help eliminate the current infection, or in the case of vaccination, prepares the body for future encounter. However, these cells don't last very long. Most survive only days to weeks. Long-lived plasma cells are very different. Instead of remaining in the lymph node, they migrate through the bloodstream to specialized survival niches within the bone marrow. Notice the image on the right side of the slide. The bone marrow isn't simply where blood cells are produced. It's also home to long-lived plasma cells that can survive for decades. These remarkable cells continuously secrete low levels of antibody, even when you're perfectly healthy and haven't encountered the pathogen for years. In other words, they quietly stand guard every single day. This continuous antibody production is one of the reasons many vaccines provide long-lasting protection. It's not that your immune system remembers the infection every morning. It's that these plasma cells never completely stop working. So while short-lived plasma cells help win today's battle, long-lived plasma cells help prepare for tomorrow's battle. And this is one of the reasons why diseases that affect the bone marrow, such as multiple myeloma or certain bone marrow disorders, can influence long-term antibody production and immune function. And antibodies are only part of the story. The immune system also creates another powerful form of protection. memory cells that quietly patrol the body for years. All right, when people think about immunity after vaccination, they often think only about antibodies. Antibodies are certainly important, but they aren't the whole story. In fact, some antibodies naturally decline over time after vaccination. That doesn't mean necessarily immunity has disappeared. The real goal of vaccination is to create immunological memory. Notice the illustration on this slide. Memory isn't stored in one single location. Instead, it's distributed throughout the body. Some memory B cells remain in the lymphoid tissues, ready to rapidly generate new plasma cells if the pathogen returns. Others circulate throughout the bloodstream, continuously surveying the body. Memory T cells do the same. Some remain in lymph nodes. Others establish long-term residence within tissues that commonly encounter pathogens, such as the respiratory tract or the gastrointestinal tract. These are called tissue-resident memory T cells, and they're strategically positioned at common sites of infection. Meanwhile, our long-lived cells continue residing within the bone marrow, quietly producing protective antibodies year after year. I like to think of this entire system as a network of emergency responders. Some are stationed at headquarters, others patrol the highways, still others are permanently assigned to neighborhoods where trouble is most likely to occur. No matter where the pathogen enters the body, some component of immunological memory is already nearby. That's what makes secondary immune response so much faster than the first. The immune system isn't starting from scratch. It's starting with experience. It's important to remember that declining antibody levels don't automatically mean vaccine failure. Memory B cells and memory T cells can remain capable of mounting a rapid, highly effective response even after circulating antibody concentrations decrease. So, what happens when the vaccinated person finally encounters the real pathogen? This is the moment the entire immune response has been preparing for. All right, at last, the moment we've been preparing for has arrived. Weeks, months, or perhaps years after the vaccination, the individual is exposed to the real pathogen. This could be an influenza virus, measles, tetanus toxin, or another infectious agent. But this time, the immune system isn't encountering the pathogen for the first time. It's encountering an old acquaintance. This rapid, enhanced response is called the amnestic response, or the secondary immune response. So let's define that term. The word amnestic comes from the Greek word meaning to remember. Quite literally, this is the immune system remembering what is learned during vaccination. So look carefully at the graph on the slide. The solid blue line represents what happens in someone who has never encountered the pathogen before. Their immune system starts from scratch. Naive B cells must first recognize the antigen. Helper T cells must become activated. Germinal centers must form. Antibodies must be produced. That entire process takes approximately 7 to 14 days. During that delay, the pathogen continues replicating. Eventually, its concentration rises above the clinical disease threshold and symptoms develop, and you possibly get sick. Now, compare that to the vaccinated individual. Notice what changes. Memory B cells are activated almost immediately. Long-lived plasma cells are already producing baseline antibodies. Memory T cells respond within hours instead of days. And as a result, antibody concentrations increase far more rapidly, often before the pathogen has an opportunity to replicate enough to cause significant disease. The pathogen may begin multiplying, but the immune system responds so quickly that it never gains the upper hand. This explains something many people misunderstand about vaccines. Most vaccines don't create an invisible force field that prevents pathogens from entering the body. Instead, they prepare the immune system to eliminate the pathogen before it has enough time to produce illness or produce significant illness. That's why vaccinated people may still become infected in some cases, yet experience much milder disease symptoms than individuals whose immune systems are encountering pathogens for the first time. The vaccine didn't prevent exposure. It prevented the pathogen from winning the race. This is why immunologists often focus on reducing disease severity, hospitalization, and death rather than preventing every infection. A successful vaccine allows the immune system to respond so rapidly that the pathogen is controlled before extensive tissue damage occurs. But what happens years later when antibody levels naturally begin to decline? Why don't many vaccines simply stop working? The answer lies in the remarkable biology of booster doses. One of the most common questions patients ask is, if the vaccine worked the first time, why do I need a booster? The answer isn't because your immune system forgot. It's because your immune system is capable of becoming even better. Think back to the germinal center we discussed earlier. That was where B cells underwent somatic hypermutation and affinity mutation. A booster dose gives memory B cells an opportunity to return to that same environment. Notice the staircase shown on this slide. The first vaccine dose establishes the initial memory pool. It creates memory B cells, long-lived plasma cells, and protective antibodies. But those antibodies aren't necessarily the very best your immune system can produce. After a booster dose, memory B cells re-enter germinal centers for an additional rounds of mutation and selection. Once again, antibodies compete, and again, only the highest affinity cells survive. This process is called iterative refinement. Each exposure fine-tunes the antibody response, increasing both antibody quality and the number of memory cells available for future protection. Notice what happens to the antibody levels over time. Without boosting, antibody concentrations naturally decline. That's completely normal. The immune system doesn't maintain extremely high antibody concentrations forever because doing so would consume an enormous amount of energy. Instead, booster vaccinations raise antibody concentrations back to and above the protective threshold while simultaneously strengthening the memory pool itself. Think of it like practicing a musical instrument. Learning doesn't disappear between practice sessions, but each additional practice session improves the speed, accuracy, and confidence. Your immune system behaves in much the same way. Each carefully timed exposure strengthens the memory established by the previous one. That's why booster doses aren't signs that the first vaccine failed. They're opportunities for the immune system to become even more effective. Not every vaccine requires boosters. Vaccines that generate exceptionally durable plasma cells and memory responses, such as measles, often provide decades of protection, whereas rapidly evolving pathogens or vaccines that induce Shorter-lived immunity may require periodic booster doses. So let's step back and look at the entire journey from a tiny injection into the deltoid muscle to decades of immune protection. So as we conclude this episode, I'd like you to look at this final illustration. Rather than focusing on the individual cell, notice the entire journey. Everything began with a tiny injection into the deltoid muscle. Within minutes, the innate immune system recognized danger and recruited dendritic cells to the injection site. Those dendritic cells captured antigen, matured, and traveled through the lymphatic vessels towards the axillary lymph node. Inside the lymph node, they activated naive T cells and helped generate T follicular helper cells. And at the border between the T cell zone and the B cell follicle, T cells and B cells met in a carefully orchestrated interaction that determined the future of the immune response. Within the germinal center, B cells underwent maturation, competition, and selection, producing antibodies with increasingly high affinity for the antigen. The best of those cells then followed two different paths. Some became plasma cells that immediately produced protective antibodies. Others became long-lived plasma cells and memory B cells capable of surviving for years or even decades. Memory T cells also established residence throughout the body, creating a distributed network of rapid responders ready for future encounters. And when the real pathogen finally appeared, the immune system didn't have to begin again. It remembered. That's a remarkable achievement of vaccination. Vaccines don't simply stimulate antibody production. They educate the immune system. They transform a first encounter into a remembered encounter. They convert uncertainty into preparedness. And they allow your body to respond to tomorrow's infection using yesterday's lesson. So when people ask, how do vaccines really work? The answer isn't found in a syringe. It's found in the extraordinary journey you've just followed, from one tiny injection to a lifetime of immunological memory. So if there's one lesson I hope you take away from today's episode is this. Vaccination is not a single event. It's a carefully coordinated conversation between anatomy, physiology, biochemistry, and immunology. Every step we followed, from the deltoid muscle to the lymph node, from the germinal center to the bone marrow, was necessary to build lasting protection. The next time someone says a vaccine simply teaches your immune system, you'll know that behind that simple statement lies one of the most elegant biological processes in the human body. So thank you for joining me in this episode of Go Beyond A&P. Learn it for the exam, understand it for life, and until next time, stay curious and keep learning.